Well, welcome to this afternoon's Safer World by Design webinar, and I'm very happy to introduce Peter Mako from the Joint Research Center in ISPRA of the European Commission. And Peter's going to discuss their approach to analyzing assay data, including the important goal of being able to extrapolate from such data that we look at in an acute sense in vitro. How do we then extrapolate such data analysis to the longer term exposure situation needed for our risk assessment? And then this will be followed by Tomasz Mohoryk, who will present then how we're taking such data from Peter and his group and going about its data management within Edelweiss Data and his presentation in an interactive notebook, which we should then be able to share with the audience to try out and also on social media after the event. So with that, I hand over to you, Peter. We're looking forward to your talk. Please go ahead.
Okay, thank you, Barry. Thank you for allowing me to present at this webinar, and I welcome all attendees. I'm happy that you are interested in our work. So I am from EUREL-EQUAM, it's the European Union Reference Laboratory for Alternative Methods to Animal Testing, which is hosted by Joint Research Center of the European Commission based in Ispra in Italy. So our role is mainly to promote and coordinate the development of in vitro use, which should be the replacement for the animal tests, and to coordinate the validation at the union level, to exchange information, keep some databases, and promote the 3 Rs and support some EU policies. And on top of that, we run a laboratory for in vitro methods in which we have cell culturing facilities, automated robot handlings for chemical preparation, cell treatment. Then we have some high content imaging platforms and also some measure devices to measure electrical activities of neurons, for example. So the outline on my presentation is here. So first I will tell you about our motivation, why we are interested in this subject and what we expected from it. Then I will show you our case study on the liver cell line Hepar-Gs. Then present the method we developed to identify a chronicity index and how to extrapolate the acute measurements into chronic toxicity. Then I will show some hints how to link it with AOPs, which are the adverse outcome pathways. And at the end, I will make an introduction to EU tox risk case study in which we apply this technique to a set of chemicals. And then Tomasz from Edelweiss Connected will show you their data management system. So from raw data to calculated data and how to get the outputs and how they are stored and so on. So first of all, our motivation was to understand better what is the role of the exposure time in an in vitro test system. It came mainly from our knowledge that in vivo measurements, the exposure time was since the beginning very important parameter. It was obvious because in vivo measurements were done in time, and it was natural to record the time when something happened, or the readouts were done repeatedly every day or every week. However, in vitro studies, the situation was and is a bit more complicated because the in vitro test systems are more complex and technically sometimes more challenging, and often the exposure time needs to be defined before the experiment itself. And then many of the readouts which are done are disturbing to the cells. You simply need to fix the cells or kill them or lyse them, and you cannot make more than one exposure time on one set of cells. So in knowledge, so we decided to apply the knowledge from the in vivo situation. So already more than 100 years ago, toxicologists realized that there is a certain relationship between the applied concentration or dose of toxicants and the exposure time. The first perhaps was the Haber, who described the effect of deadly gases used in the World War I to kill the soldiers, and he found that when he decreased the concentration by a factor of 2, he needed to increase the exposure time by a factor of 2 to achieve the same effect. So this can be mathematically expressed as concentration multiplied by the exposure time equals constant. However, later himself, but also other toxicologists, realized that this Haber's rule is just a particular case of some more general rule, which can be expressed as a concentration multiplied by the exposure time power to n equals constant. So this can be then written in a different form when the concentration to achieve certain effect equals constant multiplied by the exposure time power to minus n. And these relationships for 3 different values of n are shown on the left graph. So you see that they have these hyperbolic shapes. But when it's transferred, both axes are transferred to the log scales, you will get a straight line. This can be actually achieved also mathematically, and you get an equation for a straight line. Different other forms of this modified Haber rule exist. You can put the exponential factor either on the concentration or on the time or on the both, or sometimes there is introduced C0, which is a threshold no effect concentration, which might in some cases exist. So here are some examples from in vivo studies. On the left side, you see this relationship measured for the growth of tumors in the rats. So on x-axis you have the time, on y-axis you have the daily dose, so it was repeated daily exposure, and this relationship shows the moments when the first tumor was found, and you see that this relationship is nice linear in the log scale, and the slope corresponds to n equals 1. And from this relationship, you can define a no effect concentration. If you extrapolate it up to the lifespan of these animals, you can get the maximal dose which would be accepted. Then you can apply certain safety margins and to define the no effect concentration. Different situation is shown on the right side. Here is the effect of the warfarin, so the pharmaceutical drug, on killing of rats again. So on x-axis you have the time in log scale, on y-axis you have the dose, again in log scale, and this relationship has 3 zones. First, there is a vertical line, then there is a slope, and then there is zone 3. We are not interested in the very first. There is probably the lag between the application of the chemicals and the effects to be observed. We are interested in the middle and the third part. So why this one is bent and is approaching certain value? It means that this chemical has a threshold concentration below which the animal death is not evoked. So this is supposed to be safe to be toxic, and this concentration can be then used as a therapeutic concentration. Another example is here. It is taken from the work of Simons Delso, where they measured honeybees the mortality rate of the honeybees when they were exposed to boscalid, which is a fungicide. So on this graph you have the time in days. The blue one is the control, so the natural mortality of the bees. And when you increase the concentration of boscalid, you see that the mortality rate increases. From this graph, you can build the graph which is on the right side, where you have the log of time on the x-axis and concentration, which corresponds to LD50, so the dose which causes 50% deaths of your bees. And you can see that this nice relationships has two slopes. First one correspond to the effect of boscalid, and then it bending to down, and this is affected. This is the either the bees becomes more sensitive and they are aging, and on top of that their natural dying is manifested as well. So looking at this graph, you can define that concentration which lies somewhere here can be saved, and you can then use this concentration in your fields to be sure that you are not killing the bees. So knowing that these things works well in vivo, we decided to apply this in our in vitro test systems. So we used a Hepar-G cell line, which are liver hepatocytes, and I will show you the results which were recently published in Toxicology in Vitro. So me and my colleagues Taina Palosari and Maurice Wieland. So we used 5 chemicals we knew about the killing the Hepar-Gs, and we used a live cell imaging mode on a high content imaging platform. So the cells were kept alive over 86 hours in the control environment, and we stained the wells with an image dead green viability stains, which is simply a green staining which stains only dying cells. So living cells are not taking up the stain, only the dying cells, and once it binds to DNA, it becomes fluorescent. So by using the fluorescence platform, microscopic platform, you can see and count the dead or dying cells. So here I will show you a movie from a negative control versus a control which were treated with one of the compound over the first 16 hours. So you can see that on the left side you have only some background cells which were practically dead at the beginning, but nothing new is appearing. While on the right hand side you see the well treated with the compound, which was quite toxic, and after 16 hours a big portion of the cells were already dead. Again, here are another 2 movies where there's 2 different concentration of different chemicals were recorded. The higher concentration on the left side and lower on the right side, and you see that there is a difference in the number of the white spot. Actually, these spots should be green because it was a green fluorescent, but the black and white camera sees just the white spot. So here is a snapshot from the 96 well plate. So we had 5 compounds in 2 replicates and control wells, and 7 concentration in this case. The highest one on the top, dilution factor was 2, the lowest one here, and the negative control again on the bottom. So it was images at the beginning of the experiment, and when the exposure time was growing, so here we have 3 hours 15 minutes, you see already some cell death at the highest concentration for certain compounds, and with increasing time you see more and more dead cells. So from these microscopic images we used a high content software which identified the white spots and count them per well and per time point. And these numbers were then used to build the time and concentration response curves. So on this slide on top, you see the number of the dead cells already normalized by the total number of the cells in each well. Over time, so the signal is going from 0 to 1. 0 means everything is alive, 1 means everything is dead. And you see the time responses for different concentration of your tested chemicals. So you can see that this shape and behavior are different, not always equal. The first time point, sometimes there is a shift between the 0 time point and the first the observation of the death, and so on. The same responses can be plotted in a different way, which is probably more common for toxicologies, when you put the concentration on the x-axis and the portion of dead cells on the y-axis again. So you can see, so when you increase the concentration, everything is alive first, and then the cells start to die, and the highest concentration, everything is dead, all 5 chemicals. But what is interesting here we are showing only 4 responses, which were taken at 4 different time points: 30 minutes, 40 minutes, 30 hours, 40 hours, 60 hours, and 86 hours. And you can see that for those 2 chemicals, tamoxifen and methylenbis, those concentration responses are practically overlapping. So nothing significant is happening when the exposure time is increasing. While for these 3, you can see that the responses are moving to the left. So at the beginning, the response is at the high concentration, but as the time is running, the response is shifting to the left. Here the question is, how far would it move if we could keep the cells in the same conditions longer? And would it move? Well, that's the questions we tried to answer in this experiment. So we plot this graph, which is the this ISO effect graphs, so similar to the in vivo studies which I showed you before. So on x-axis we have the exposure time, here is in days, and on top it's in hours. And on y-axis we have the concentration IC50, which corresponds to the concentration which at given time point evokes 50% cell death. And so at every time point we fitted a hill curve to the observed data, and the fit gave us the IC50 value, and we have plotted it here. You can see that the first 2 chemicals which I showed you before, which were practically overlapping, so here these relationships is very horizontal. In the case of the methylenbis, it's practically horizontal. In the case of tamoxifen, is only slightly decreasing. For another 3 chemicals, we see that the decrease with the exposure time is more significant, and the highest is for the rotenone. So then we applied the modified Haber rule, which is shown here, and fit to those data starting from roughly 1 day, so somewhere here. So we fitted only the end linear parts of these relationships, and we got these numbers of n. So for this one it was 0, for tamoxifen 0.12, and so on, and the highest one was for rotenone 3.1. And now the question is, can this fitting be used to extrapolate to longer exposure times which are outside of our measuring windows? And how far could we go? Where should we stop? So that's the question we asked ourselves. And then we suggested to combine in vitro knowledge with in vivo knowledge. So it is known that in vivo human hepatocytes is in healthy people with healthy liver usually survive around 200 to 300 days. So we plotted this time as vertical lines in this graph, and we extrapolated our observation up to these points. And the intersection with 300 days gave us the chronic extrapolated IC50, and the numbers are shown in the table here. And on top of that, we suggest to call the factor n, so the exponential factor in this modified Haber rule, as a chronicity index. So if n is equal 0, the extrapolation is practically horizontal. So whatever the exposure time is, the concentration IC50 would still be the same. So this chemical is not the candidate for chronic toxicity because it seems that the effect is independent of the exposure time. While when n becomes bigger, for example, n equals 1, you get the Haber rule, then the effect due to the toxicant and the chemical and effect due to the exposure time both will be equal. So the effect will be multiplication of both. And then if you further increase n, so for example, in the case of rotenone, which is n equals 3 and something, you can see that the extrapolated IC50 value is far away from the value which was measured in the short-term in vitro experiments. So n will be an index which would be easily below the extrapolation between two measurements, or between the measurements and what you expect. And then lower value means lower chronicity, and higher value would mean higher chronicity. So I go further. So in the image below, I was showing just the extrapolation of the IC50 value, but you can extrapolate your full concentration time responses, so which is shown here for all 5 chemicals. So on top of 0.50%, we extrapolated also 0.2 and 0.8, which corresponds to the cell death 20% and cell death 80% for all 5 chemicals. So in this way, you can extrapolate the point of departure and get the chronic point of departure, and then you can apply some safety margins, and you can get even the no effect concentration, which is interesting for some fields of toxicology. So what is behind of this behavior? So we have studied further in the literature, and we have found very interesting work which was done quite long ago by Drucker and Kumpfmuller. So in the book entitled Doses and the Wirkung, which is written in English, but many of the most interesting parts have been published by Tenekis in several papers. So here I show just this one, from which comes this very nice and interesting table with some mathematics. So this is way how they were thinking what is behind these different shapes, and they supposed quite a simple system, which they describe by certain terms, but I prepared an updated version which is based on the AOP terminology. So they suppose the very simple AOP, which consists only of 2 events. The first one is the molecular initiating event, and then one key event. And the molecular initiating event is the bonding of your toxicant to your target molecule. And then the key event is a kind of damage which is developing in the cells or in the systems before some measurable effect is observed. And then they supposed the molecular initiative event. They suppose two extreme cases. Either it is this dissociation is possible. So if you remove your toxicant in a finite time, everything will be dissociated and the initial cause of your effect will disappear. So in this case, the number of the targets to which the concentration, the molecules bounds will be proportional to the initial concentration. And then they supposed a key event, and again two extreme cases. Either there is a full recovery or no recovery. If there is a full recovery and you remove your initial cause, the system will recover and will get healthy again. So it means that the effect at a certain time point is proportional to your initial concentration, or to the bone target concentration, which is proportional to the initial concentration. So you get this relationship. Effect is proportional to the concentration, which corresponds to the modified Haber rule with n equals 0. So the horizontal line in my previous graph. Then if we suppose another case, if the molecular initiating event is permanent, so there is no way for the dissociation. So then you need to integrate your concentration which launches the molecular initiating event over time. And this becomes the input for the key event. And if here we suppose that the recovery exists, so then the effect will be proportional to the input of the key event. So it's simply the integral of the concentration over time. And if the concentration and at the place of action is constant, then you get these relationships, which is the modified, which is the Haber rule. So n equals 1. You then take the opposite combination. So the MIA is not the permanent, but there is dissociation available. But now the key event is of permanent nature, so there is no recovery available. Then you will get the same results as for the previous case. So again, effect proportional to the concentration and time. So n equals 1. And now, if you suppose that both the MIA and the key event are permanent, so there is no way for the recovery and no way for this dissociation, the number of the targets bound to the molecules will increase with the time, so with the integral of the concentration over time. And then again, you need to apply another integral over this integral, so you will get double integral of the concentration over time. So in the constant exposure scenario, you will get this relationship, concentration and time square, which correspond to the modified Haber rule with n equals 2. So, but here I have to stress that this holds true only if the effect and the binding is a linear function of the concentration at the place of action, which might be the case for some chemicals, but we cannot expect that this is a general situation for all chemicals and all AOPs. But if we suppose that it is true, so it's linearly proportional to the concentration, then we can find these interesting consequences. So imagine now that you have not a constant exposure, but a dynamic exposure. So the opposite of the constant is pulses. So imagine that you have 3 pulses like that of your toxicant at the place of action. So it binds to the and you suppose the first scenario, so everything is reversible, the binding and the damage, which the binding causes. So your effect will follow the concentration. Here is for demonstration, there is certain shift because the effect is always lagging a bit behind the concentration because the processes are not instantaneous. There is always some time needed to evoke and to bind and to recover and so on. So this is effect which is causing the chemical in cells, for example. And if you now imagine that there is some threshold needed for this effect, for the cumulation effect to cause, for example, the apoptosis, you will get this situation that only the third, the highest pulse will evoke the cell death. So this corresponds to the Cmax scenario. So the amplitude of your Cmax, so amplitude of your chemical at the place of action defines the amplitude of the effect. Then let's suppose the second case when n equals 1. So again, you have 3 pulses of the chemical at the place of action, and then you apply the first integral over time, and you will get something like this. So the first pulse will cause certain effect level of damage, and then when the concentration decays to 0, the effect will still be present until the second pulse, which brings it to a new level, and the third pulse brings it to another level. So if there is no recovery possible or the binding to the receptor is permanent, you will be in this situation. And then you can imagine certain threshold. So once you go through the threshold, you will get the measurable effect, let's say the apoptosis. So here the effect is given by the AOC, so area under the curve. And the third scenario for the n equals 2. So first imagine you have the first pulse of the chemical, and you make the second integral of the concentration over time. So it means you first calculate the area at every time point, and you will get how the area is increasing with your time, and then you apply a second integral. And the result is that it will be increasing at a certain, well, rate during the pulse. And then when the concentration drops to 0, it still will keep increasing. Then when you apply a second pulse, you change the slope of this increase. And you apply the third pulse, you change again the slope. The higher the amplitude, the bigger change of the slope. So, and if you imagine here some hypothetical threshold, the first pulse would cause, then the effect will appear at this time point. But if you apply the second pulse, the effect will appear earlier at this time point, and the third pulse bring it here. So that's the consequence of this double integral. So what is important to keep in mind that mainly in the third and second scenario, if the initial conditions are non-zero, it's impossible to make some assessment because you don't know in which state your system is, and if you don't know the pre-exposure, which is particularly difficult to protect animals and people if you don't know what their history is. Now you can imagine what would happen if the n is not equal to 0, 1, or 2, but somewhere in between. So it's, for example, it would be 0.5. You would get something similar to that, but this decrease of this effect to 0 will be slower, but it will be still decreasing. And here, if it would be somewhere between 1 and 2, so then it will be something between these two scenarios. So this is the main things which I wanted to show you. Now I will show you another experiment. So having these things in mind, we decided to apply the same principle in the Eurotox EU tox risk case study. So it was the consortium of many laboratories which collaborated on developing some approach to quantify the the toxicology. And me and with my colleagues from the unit, we applied this approach on a list of chemicals which were selected in this consortium. As the number of the chemicals was much higher than 5, there were 76, plus we have some other chemicals. We decided to use a faster approach. So instead of cell counting and high content imaging, we used another staining which measured the overall cell viability per well, and we used the well-paid reader. And as there were many chemicals, we used QHTS approach. So we had 8 plates. Each plate has one of the concentration. So the highest one in plate C1 and so on to C7. Then we had some negative and positive controls and 3 replicates of each condition on plate. And then we had 3 biological replicates. And those data were exported, and the same analyses were applied. So here I will just show one example. So for certain chemical, we got these responses as a function of time. So this time, this is reading. So higher signal, more cells are alive. So the highest concentration is the red one, and so on. It was decreasing. And then we applied the normalization using negative control or the MSO control. So those signals were divided by the observation in the DMSO control, and we got these concentration responses. So the gray are the variation in the negative control, and the colored are the observation. So here everything was alive, and here we see that the cells were dying as a function of time. So from those we plot 2D heat map graph with the time on x-axis, concentration on the y-axis. Yellow correspond to living cells, blue to the dead cells. And by applying the Hill fit on each time point, we got the IC50 values, which are the red dots. And then we apply the modified Haber rule fit. And for this particular chemical, we got n equals 0. So this is not a candidate for chronic toxicity, but only for acute toxicity. So now I will thank you for your attention, and will pass the floor to Tomasz from Edelweiss Connected, who will show you those data from the EU tox risk case study, how the data were exported, analyzed, and so on. So thank you very much for your attention. And Tomasz, the floor is yours.
Thank you. Thank you very much for your presentation. It was very, very interesting. Now I will share my screen. I hope you can see my screen now.
Tomasz, can you also come back on camera?
Yes, sure.
Thank you, Peter. Very interesting talk. I just want to remind the audience as well, because I saw a lot of interesting points and possible questions you were making there. Please submit some of your questions through this question box, and we'll, after Tomasz shows the notebook, maybe we can have some then discussion from your questions. Go ahead, Tomasz.
Okay, so as Peter mentioned, the last experiments that he showed, they were part of EU tox risk project. And in EU tox risk, there are many different laboratories, partners involved, and each of them ran different assays. And we were then challenged with how to organize data so that it will be useful later on for integrated analysis, for comparison between different assays, and so on. Now I would just maybe start at the end. So to show you an example, what is possible if you have a good data management, so that you can be sure that, for example, all your data sets that you created in the lab are stored in a cloud-based database, and that they all conform to some rules, that they all have certain fields that are common to all of them. So, for example, also this allows you to make data easily shareable with anyone, so that you can be sure that everyone is looking at the same version of data into their analysis. So, for example, this is a notebook based on data provided by JRC, data that Peter also showed briefly. And here, basically, user can pick an experiment. So each experiment generated a separate data set, and we will see how data was then organized. And for example, by picking a data set and then selecting a compound, all these lists are automatically updated from the cloud. You can immediately see, for example, I can pick a different compound, and all the visualizations that are automatically generated, they are updated immediately. So, for example, if I can pick compound number 2 because it has interesting response, I will immediately get, for example, the results for that compound. This is the average response. These are individual replicates. So what we are seeing here is dose versus concentration and time. So this relationship that Peter showed in the presentation. And after that, okay, here we see heat maps. We see some interesting patterns, and then we can also make individual dose response relationships for individual time points. For example, we can select time points that we are interested in, and they are immediately displayed. And what we did after that is we also fitted each dose response at a given time point with this Hill equation to get IC50 values. So for each time point, we obtained IC50, and then we determined chronicity index as this by fitting this equation. So IC50 as a function of time to a certain exponent. So the advantages of having data in such database and making it available through web requests, it means that you can make such notebooks for visualization or for analysis to run in the browser. So you can easily share such notebooks with anyone, and you can be sure that they see the same thing and they can run or modify for themselves. So it's not that you run your analysis locally and then you transfer just graphs, but you can transfer basically everything together. So behind such a notebook is also some code that was needed to generate it. This is particularly JavaScript, but everything starts here where you basically access data from the database, from the cloud. And after that, you basically just reorganize data that you accessed to show on the interesting part. Now, as I mentioned, in EU tox risk, there were very many laboratories, and each has different assays. And now the challenge was how to organize all the data sets so that they are all available through as a web service, so that you can access them from different tools. Like this is just one example, observable notebook. You can also access data from Python, R scripts, from Excel. So in EU tox risk, we decided to build a certain template that each data set that was produced in the project had to conform to. So for example, each data set has, for example, this one part where all the measurements were stored. So each row is a single measurement. It has defined some sample ID and compound concentration, then endpoint data. So what was the raw measurement, and then what was the processed data. And this part is then accessed through web. But it's this now that what we are looking now is just the data, so the variable, the measurements. But each data set also has metadata that is also crucial, and metadata describes all the parameters that are relevant for the assay as a whole. So like what was the cell type and what was the organ or organism and so on. And this data is also stored in a machine readable format. Here we used JSON format, which is very flexible. So basically you describe all your assay, all your metadata in the form of key value pairs. And also, for example, compounds are also stored here as a list where each compound has then defined all the relevant identifiers, or for example lot numbers and so on. And all this data and metadata, they are available through web, so that, for example, in this notebook which runs in the browser, immediately when I make a change, for example, pick a different assay or pick a different compound, every time a request is made to this cloud-based database, and that part of data, the relevant part of data, is extracted and displayed. So the challenging part that we had, for example, was to, this is example of raw data from the JRC. The challenging part was then also to create customized workflows to convert raw data. So these were measurements produced by the instrument, how to convert all this data into a format that was agreed for the whole project. So, for example, these plate layouts were converted then into Excel, where each row contained measurement of one well, and everything was defined. For example, okay, this is row of the plate, column, plate number, and what was the content of the plate of the well. So what was the compound concentration and what was the measurement. So all this information was for all the assays in the project, it was converted into this format, and this allows you then to build such notebooks or any kind of workflows in maybe Python, R, whatever, because then you can be sure that all data sets that you are trying to access, they are in a particular format, and the data is then really useful, and you can make such visualizations and interactive notebooks. So this is briefly about the data management part behind the essays that Peter showed. So with that, I think I can finish, and if there are any questions, I'm happy to answer.